All right, so there's another way. Instead of taking now purified protein vaccines, instead of having the whole virus, when for vaccines that are using purified protein, they're trying to actually get one part of the the virus. So instead of the whole virus and inactivating the whole virus, you're just going to try to take one part of the virus and purify it and inject that as a vaccine. Now, this is an interesting bit of history because this was a lecture and let me bring up a video.
Now this is a receptor for that enzyme. So again, that's why high blood pressure is linked to increased risk of complications from COVID, right? Because it's binding to that receptor. So if I was creating a vaccine, what would I target? So I would actually target this spike. Why? Because this is the one that's actually growing, glomming onto this ACE receptor right here, right? So if I give a vaccine that's trained to bind to the spike receptor, it's going to block these spike proteins from binding to this receptor, thereby preventing these cells that have the receptor from being infected. So if you're able to opsonize, create antibodies against the spike protein, this prevents SARS-CoV-2 from binding to the receptor, hopefully preventing all the complications from COVID-19.
Yep, so that was last year. And that's the strategy most people went with. I wish I could be that correct all the time, but that's what people were going for. Despite all the vaccines that are currently working, they're targeting the spike protein. So they're not using the whole coronavirus. They're trying to train your immune system against that spike protein that the coronavirus uses to actually get into your cells. Now, let's talk about another vaccine that's already an example of this. So some of you might have already had Gardasil or Gardasil. And what is this trying to prevent? Well, it's trying to prevent HPV. And actually HPV are multiple types of viruses. So Gardasil tries to protect against several of the subtypes of HPV. So what happens here? So this is what HPV looks like. So HPV is another virus. It has all these proteins. So HPV has DNA. This is how it's able to replicate and reproduce itself. Now, this is the really interesting thing with Gardasil is that what they do is have all this part right here, the outer covering of it. What they did is try to make this protein, and the cool thing is that this capsid protein that surrounds that genetic material, so this is the surrounding you see right now on the surface of the HPV, is that if you can have this purified protein, it assembles and forms this ball again. But because it's just protein, no DNA, the inside of this ball is hollow. So you kind of like have a blank, you have a dummy, you have kind of a shell of the original HPV. So the cool thing is that you have something that looks like HPV, but it's a mannequin. It's hollow. It doesn't have any of the reproductive capabilities of HPV. So that's the whole principle behind Gardasil, is you make this purified protein, and then you have something that looks like HPV to your immune system, but it trains it to recognize these proteins on the outside. And then when you actually get exposed to HPV, then your body is able to fight against that. And it's very important, again, so why do we give that? And the thing is, like, why is it controversial? Well, the thing is that this protein it causes a very strong immune response. And I know, and if you take like some of the public health, like Dr. De La Cruz's lecture, like often people say, okay, why are we giving this to teens? Why are we giving this to someone who's young? And the thing is, like, we're not encouraging, by giving the vaccine, it's not encouraging sexual activity at that age. It just happens to be the age where it's most effective in training your immune cells and generating a high antibody titer against HPV. So that's the biggest thing about the age for giving Gardasil, is where it's most effective. And that's why now it's, I think, approved up to age 45, but that's why it started off in that age range, because again, that's when the immune system produces the most antibodies against this HPV strains. So how do you make viral proteins? Well, what you have here is your coronavirus, right? So what you want to do for the viral protein vaccine is just make the protein without the whole virus. So thing is that we have to go back to cell biology. We have to go all the way back to the cell chapter. And if you're not if you haven't taken a cell biology or any biology class, I have my cells lectures on my YouTube channel as well. So DNA has information to make every protein in your body, structural proteins that make it the shape of your cells, your extracellular matrix, and also the proteins needed to divide cells and undergo mitosis. Now, how do you make a protein from DNA? Well, again, if you remember this from last year, DNA, you do transcription, and DNA is your permanent record, and you have genes on DNA. Then you make copies of DNA called mRNA. Now, these mRNA copies undergo translation, and they become proteins. So remember my little mask analogy, same workflow there. So your protein is your final product. Now with the coronavirus, what it does is that your coronavirus, the SARS-CoV-2, does not have DNA. Its genome, its instructions are actually mRNA. It cuts out the DNA step completely. So remember, nucleic acids contain information. So again, for your cells, your DNA has information to make every protein in your body, all the proteins that make the structure of your cells, and also the proteins needed to divide and replicate your cells. For viral, RNA, or DNA, so viruses, they can either have DNA or RNA genome genetic information. For coronavirus, it has RNA, it doesn't have DNA. So what it has is make every protein in a virus. So with that viral coronavirus mRNA, it knows how to make every protein. Also structural proteins as well, and proteins needed for viral replication. So it has that information. Now, how can we use that information? So this is all the genetic information for the COVID-19, the coronavirus, right? So you have all of these proteins, but what do we want to do with a purified protein vaccine? We want to take the spike protein that we mentioned earlier. So what we want is just take the spike. Don't worry about the rest of this, because if you take the spike protein, the part of the RNA and genetic information that only encodes the spike protein, and leave out the part where the instructions that allow a coronavirus to replicate, so you're going to get rid of the reproductive part of coronavirus, you're only going to get that structural part of coronavirus. You're only going to take that spike protein. So what you're going to do, take that genetic information, put it in lab-grown cells. Again, this is not putting it in a human. You're going to put it there are many cell types you can use to take genetic information, and then you're going to take that viral mRNA or the equivalent DNA copy of that. So the cool thing is that you can actually go from mRNA to DNA in the lab. So you can take that genetic information either in DNA form or RNA form, put it into cells, and then the cells will start cranking out that spike protein. So you go from nucleic acid to a protein. And then you can load up, again, it's just a protein, it's not the whole virus. So you're going to take this purified protein, you're going to harvest it from the cells, put it into a vaccine, and then inject this protein. Will this cause coronavirus? No, of course not, because again, you don't have the rest of the cell. You don't have the replicate, one, you don't have the nucleic acids, you just have protein. And two, you don't even have any of the reproductive machinery for the virus as well. So what happens there? Well, you're injecting the proteins, and then remember, if you haven't, this is why it's important to go back to the adaptive immune response lectures and cells if you haven't gone through already, but to review, dendritic cells. So again, they're the little feelers of your immune cell. They sample the surroundings. So what if you inject this purified protein? Well, these dendritic cells sample the surroundings, and hopefully they'll pick up some of the spike protein. They're going to process it, break it apart, and what do? Well, they go to your lymph nodes, they present it to naive T cells. So here we have a dendritic cell, and remember that these are professional antigen presenting cells. Professional APCs have exclusive use of MHC class II receptors, and this is what they use to put these antigens on and display it to other cells. Now, what if you have a T cell, a naive T cell that was, again, you generate many combinations of naive T cells with different types of T cell receptors. What if a naive T cell that has a T cell receptor that also recognizes the coronavirus spike protein so what happens is that this T cell is able to recognize that coronavirus protein. And now this dendritic cell is providing evidence to this naive T cell, hey, I found this antigen, do you recognize it? And the naive T cell is super stoked because it's like, finally, it's my moment. It's go time. I have finally got my number called. It's time to replicate, become a helper T cells, cytotoxic T cells, and train other cells as well. And remember that not only naive T cells, but also B cells can present antigens as well. So here we have a B cell, and it's naive right now, but what happens is that this B cell is not activated yet, but now you have all the purified protein from that vaccine. If this B cell has B cell receptors that recognize that spike protein, then it can process it, bring it inside the B cell, and also present it on its own MHC class 2 receptors. So B cells that recognize the spike protein can now be activated. So here we have a B cell that's able to recognize the spike protein or its antigens, and it's presenting it on an MHC class 2 receptor. Here we have a helper T cell, and this helper T cell is like pretend like this helper T cell was an offspring from that naive T cell that recognized the spike protein earlier. So these two cells are now communicating. They recognize the same antigen. They're double checking each other. So they're pretty darn sure that this spike protein is here somewhere. What are they going to do? Well, this helper T cell, remember that B cells, even though they bind their antigen, they need another step. They need another push to finally become a fully active plasma cell. This helper T cell gives it that extra push. They're super happy because now they know what's up. This is one of those images you can hear in your mind. So now the B cell is fully activated because, yeah, it found the antigen and it found a helper T cell that recognizes the same antigen. That helper T cell is going to tell the B cell, hey, now you can finally crank out those antibodies. So purified coronavirus protein vaccines, this is the whole principle behind that. Now, the Vector Institute in Russia, they've come out with one, and I think it's approved in just Russia, and I think that's Turkmenistan. So that's probably why you haven't heard of the Vector Institute vaccine. There is an American vaccine being developed, but it's not yet approved in any countries yet. So yeah, so vaccines relate to proteins in our body. So yeah, it's not things that you could generate a vaccine. Well, with the Chinese and Indian vaccines, they're trying to just use the whole virus itself. The protein is more of like a refined target, and they're trying to prevent and again, this is another difference between why the Chinese and Indian vaccines, I don't think I've seen them ever I think the Chinese one claimed it was 90 something percent effective, and then Brazil tested it and they said, oh, it's only 50%. So that's one big difference in strategy. It's not, you don't have to target a protein, but the protein is, again, just a surface molecule. It's not actually the reproductive part of the virus. It's only a part of the virus. And this is why, so that's a big difference in the strategy.